Okay, welcome everyone. We're going to go ahead and get started. Good morning, and here at the University of Utah. On behalf of Will and George and the rest of the organizing committee, I want to thank you all for coming. We have a truly exciting day of programming planned on some from Population Health Science here at the University of Utah. And I've had the privilege of being on the organizing committee for last year and this year's symposia. And for some background on the event, the Frontiers in Precision Medicine Symposium is such an important event to us here at the University of Utah because it catalyzes our focus and initiatives around personalized medicine, genetics, ethical, legal, social issues, and bioinformatics approaches to achieving the triple aim in healthcare. And the vision of this event is to bring people together precision medicine forward, and what are the ethical, legal, and social issues we will face. The inaugural symposium was held in 2015, and over the past 3 events, we've discussed these issues as they relate to topics around pharmacogenetics, cancer genetics, payment and insurance of precision medicine, health disparities and race, big data, prevention, shared decision-making, population health, among others. And this year, we chose to focus on the role of assembling large cohorts and artificial intelligence in precision medicine. As you all know, the NIH-funded All of Us research program has begun enrolling participants in a historic effort to collect data on over 1 million or more people to accelerate precision medicine and ultimately improve health. And at the same time, the president signed an executive order last month meant to promote the development and regulation of AI, or artificial intelligence, which, as you know, many experts believe will define the future to, of course, healthcare. So some of the questions we will focus on today are, what are the ethical, legal, and social issues in assembling cohorts for precision medicine like the All of Us research program? Can AI in healthcare lead to higher quality of care at lower cost with a better experience for both the patient and the provider? Can AI improve healthcare decision-making, gene discovery, and aid in the phenotyping when we generate large cohorts for research? Today, we are very lucky and excited to host several leaders in the field on these topics. We have a leader in machine learning in healthcare who was named to Forbes top 30 under 30 science innovators. We have the director of worldwide AI policy for Microsoft, and an international thought leader in the ethics of genetic research and the translation of genomic technologies into clinical care. So please take advantage of this opportunity to meet and mingle with people you've heard about but may have never met. I really hope you enjoy. PREC-MED, so UU Precision PREC Med. So please help us spread the word of today's conference by live tweeting. And now I'd like to invite one of my dear colleagues and mentors, who is currently the Interim Associate Vice President for Research and the Director of the Program in Personalized Health at the University of Utah, to kick off the session. Thanks, Will.
Thank you, Adam. Over the past 20 years, our keynote speaker has established a stellar scientific and leadership career in both academia and biotechnology, and on is currently Chief Scientific Officer and Senior Vice President of Discovery, Pharmaceutical, and Translational Research at BioMarin, a California-based biotechnology company focused on rare diseases. Dr. Carden received his PhD in behavioral genetics from the University of Colorado, and his research efforts have focused on developing and applying multiple statistical methods and software to gene discovery in complex chronic disorders. He's held academic at the University of Washington as part of the Fred Hutchinson Cancer Institute. He joined biotechnology industry in 2008 and quickly became a senior vice president for the biopharma giant GlaxoSmithKline, and he moved to BioMarin in 2016. He has been recognized for both his United Kingdom and in the Association for the Advancement of Science. Dr. Carden has been a key leader in the All of Us initiative, both on the Scientific Advisory Board and since 2015 as co-chair of the Genomics Working Group. So please help me welcome Dr. Carden to give this morning's keynote speech.
Well, many thanks for that very kind introduction. It's a real honor and pleasure to be here. As Will noted, I'm a geneticist by training, and it's a bit daunting to come to Utah, which I personally see as building maps and all the diseases that we've been able to map genetically. So it's a pleasure. It's a real pleasure to be here. Will gave me a fairly open remit in terms of describing large cohorts, and one could take that in a lot of different directions. I thought I would take it in the direction that is nearest and dearest to me, and so I want to set that up for you so you know what's coming. I'm interested in the better treatments for rare and for common diseases. I think precision medicine follows from that if we do that well, rather than trying precision medicine for precision medicine's sake. If we can find the causes of disease, find a way to treat them, to make new medicines and treat them. That's where I'm coming from. That's my interest, my scientific interest. Academically and industry, it's all the same. I'm going to step back from large cohorts for a moment and motivate the problem on why we're even interested in that from that context of translation. When you're old and gray like me, you can remember at least in 2019, has had precision medicine in the title or near it in somewhere. AI and machine learning are right next to it. Big data, population health. But it's useful to know that, so what I put there are just the, I don't know if the mouse works here. Back in, cystic fibrosis was cloned back in, I think, 1989. So it's 30 years coming where we're just now getting treatments for that. What you see in the clinic right These things were found 30 years ago. And yet we as a community get very excited about precision medicine or about AI or whatever, thinking it's going to deliver tomorrow. Right? We need to realize this is a long haul. As a young professor, I thought I invented it, but there's a whole field around this. It's called the hype cycle, and it is pervasive in IT, as I understand it. So what happens there? You come up with some new technology. Everybody gets very, very excited about it. It doesn't deliver to the initial promise. Everyone runs away from it, and just about the time everyone's gone, it delivers. Okay? I don't think you could describe the history of human genetics better. That's exactly what has happened. We got very, very exciting Human Genome Project. We ran up to about 2000, 2001, thinking we had industrialized genetics to find new medicines. Everybody left. it crashes down to about 2006, 2007. It comes back. It's transformational in oncology right now. It's coming in rare diseases, and we're coming back. So that's the hype cycle. But we need to keep that in mind, I think, as we approach the translation of basic findings to research. Now, now, in terms of basic findings, is stunning. So you go back to the CF or the Huntington's case. I was not part of the gene finding approach for Huntington, but followed on from it to look for modifiers of Huntington's. And Huntington's was mapped by this really rich Venezuelan pedigree, which Utah people will know pedigrees very, very well. And that pedigree would go around about half the room, the picture, just the drawing of that pedigree. It was so complicated to draw on that. And it took years and years and years to find the genes and to try and do something with them. And now, 30 years later, we're finally getting them into the clinic. Now, largely because of sequencing technologies and initiatives like the Undiagnosed Disease Network, if you look at it now, disease genes for monogenics or for rare diseases are being identified at the rate of 3 per week. 3 per week versus 30 years ago. So the question is, what are we going to do with that, right? That's where we really are. It's not about finding these. We can find the causes of these things. It's what are we going to do with that information? Now, fortunately, for rare diseases, we're in a pretty good situation where the curve, those are both just showing upticks in approvals or in applications to the regulatory agency. So for rare diseases, the basic research is coming along beautifully, and we've got a regulatory agency that is adapting to those findings. Picture's looking pretty good there, and really promising for rare disease. Common diseases, when I was still in academia, back in about the 2000s, early 2000s, a number of us and a number of Utah investigators, Lynn Jordy was part of this, very much so, would have really dynamic debates on whether we knew of 10 genes for common complex diseases, or 8, or if you were really bold, you might say 15. And we would debate whether they were robust and whether they were real and whether they would hold up, and it was ApoE for Alzheimer's and others. And so we didn't know, firstly, if we could find the genes that cause common diseases, or even if they existed. Right? So on the background of that, my first exposure to large population cohorts is in about 2001, 2002. I was in the UK, and we wrote a grant, a pilot study to the Wellcome Trust, which is the second largest biomedical charity and very, very good for funding sort of harebrained ideas. And a couple of us got together, and we detectable genes for common complex diseases, right? And this was called the Wellcome Trust CaseControl Consortium. This slide kind of got bungled up, but what we did, this was emerging technologies, is we didn't know if there were genes out there. So we went to the whole country of the UK, fell out that had at least 1,500 samples in any disease. And the Wellcome Trust said, you pull this together, we'll write the check. Okay, so everybody came together, whatever that check is. Everybody came together that had common diseases, because we didn't know if there were genes for hypertension versus genes for type 2 diabetes. So we had to sample them all. And my initial proposal, because the technology was so bad, was to do 1 chromosome of the human genome, because that's all we thought we could do. By the time we got funded, the technology had emerged, and we got to do the whole genome. So assembling that collection was my first experience. Remember, we had 10 genes for common complex diseases, and the results of that study, where these are just pictures of statistical hits, and just look at the green things that are large. Those are I think we had 21 in one study. So doubled the history of the field in one study and demonstrated that these genes could be detected, right? My personality is such that it's instantly knowing that we could find them, I was bored and moved on, and that's why I went to industry, because that was my career path. Just to give you a hint of where we are today on this, Take coronary artery disease, that was 2007, and that was the finding. It was a very interesting one because it's not even in a gene. Genetics always surprises us. It's just some location. Here's where we are today for that same. So we went from, in 2006-ish, 10 genes for common complex diseases. Today, the genetic variants, there are about 6,000. Field completely transformed, right? So one would think that industrialization of healthcare, where are all the drugs coming out? Where's precision medicine? We should be doing a victory lap here of all the findings and the new medicines that we've got, but we haven't got there yet. And in fact, if you turn to the industry side of things, these findings are paralytic. The Huntingtons and the CF and Duchenne's, to having so many we don't even know where to start, which is absolutely the case today. There are 200 plus genetic sites associated with inflammatory bowel disease. What do you do with that? Where do we start with that? Where, in terms of precision medicine, in terms of starting, it's really, really complicated. So we've gone sort of a situation where the cup runneth over, and we've got so much to deal with. Findings, people will often say the effect sizes of those are very small, which is true. So any one of these does not, it's not a Duchenne's effect. It confers a very little risk. But the effect size for genetics do not equal the effect size for drugs. I put an example up here that Will know very well. HMG-CoA reductase is the gene behind statins. PCSK9 is a more recent one. PCSK9 has a massive genetic effect in humans. HMG-CoA reductase has a microscopic genetic effect. They're turning those into medicines. Both have very, very large effects. So genetic effect size does not equal potential drug effect size, which I only diverted to emphasize because all those GWAS loci, there are 6,000 of them and they're all small, but that doesn't mean they're inconsequential. That HMG-CoA reductase you wouldn't pull out of, that would be buried in the GWAS thing, and yet statins. So we got to take them seriously. We got to take them all seriously as potential drug targets. Why does this matter? In the drug discovery domain, I've highlighted rare there, but if you take all the history of medicines that have been successfully or unsuccessfully attempted, there's some link to disease, and those that don't. Right? Most of them actually don't, because they're historic. We didn't know the genetic basis of disease. But take those that do and those that don't. The chances of success in drug discovery are doubled for common diseases just by choosing to start with something where you know nature already showed you there's a real link to disease. It's as simple as that. Double. In fact, it's the best predictor of drug success of anything that we know apart from already drugging the same target and coming back with a because these are monogenic and they're really big effect sizes and they work. So that's really impressive. Just use the genetics. So you would think in this day and age, we're 10 years into this now of having discovered 6,000 genes, that the whole world would be using these findings. It's kind of interesting, actually. This is a plot, a clumsy one, and I've anonymized it as to not offend any of the big pharmaceutical companies. But on the x-axis there are all the big pharma. So GSK, where I used to work, is in there. Amgen's in there. Roche is in there, and all that. The blue bars are the percentage of things in their pipeline that use that genetic information. So we know it doubles your chance of success. We know it works. We know it helps. They sit around about 15% of the things that they spend billions of dollars on per year even have bars. So the red bars are things for which they've got something in their pipeline and it has a genetic association or a genetic defect and it's linked to a disease that's not the one they're working on. Really, really interesting. So the genetics is pointing in this direction and they're going in this direction. That's something we can capitalize on, and I think we're going to come back to this, but that's where the cohorts come in, because you've got to have broad phenotypic information, and just collecting information or collecting type 2 diabetics when in fact the genetics are pointing over to something very different is not going to get you there. I think the only way you're going to get there is large cohorts. So I think this. So in my way of thinking, we're actually in a really positive time. So I start with the hype cycle, and it looks all negative, and I say, you know, no one wanted to talk to us back in the early 2000s because we hadn't delivered. Now, I think from a translational perspective, it's up to the community. It's up to the medical community. It's up to the genetics community. There's a big computer science component of this to figure out how to do this and how to make these translational findings, because all the pieces are there. We have all the genes. It's great to go find more genetic discoveries, but we've got 200 plus genes for Crohn's disease or inflammatory bowel disease. We should be able to make some starts in there. We've got that many again in type 2 diabetes. Last I checked, there was 100 plus in schizophrenia. We've got plenty to work on, and we've got all the rare diseases. What's happened recently? That aren't in drug discovery won't know this, but there used to be something called this concept of the druggable genome. So we have around 20,000 genes, but only some of them are really amenable to the technologies that are available in industry, chemistry approaches to go make a molecule that can get into a cell and do exactly what you want. Only a small number of those are amenable to that. But now we have gene therapy, which we can absolutely replace things in. We have oligonucleotide therapies. We can go modify RNAs. That whole concept of the druggable genome is kind of gone. We could probably go after just about any gene in the genome because we have the tools to do it now. So there's no excuses. We've got the genetics behind the disease, and we've got the pharmaceutical tools to get there, which I think is really, really, really exciting. And on top of that, we have one of the breakthroughs, I think, scientific breakthroughs of in my lifetime with world used to have to just sample and sample and sample people until you detect someone with the mutation you're interested in, and now you just make it. And it's years of savings for just about any basic research program. Completely transforms basic research. Whether it's ready for the clinic, question mark, but from a basic research perspective. So we have the genes, we have the tools, and I think the third leg of that stool is getting them into cohorts. I think we've got to get them into, and the cohorts will unlock those tools. So to me, this is the right time. There is no excuse to fail. We've got everything sitting in front of us, and it's up to us to figure it out, how to do it. Now the problem is, we don't know how to put it together, and nobody does. Which is why you have pharmaceutical industry still doing things the way they have for 30 years, because we don't know how to use these pieces. I think this is a fascinating time, because we used to say, oh, if only we had a little bit more data, or if only we had a means to do this. We can't say that anymore. It's up to us to figure it out. So that's where we are. So for those of you who are thinking, oh, can't you pharma guys just go figure it out? Because a lot of people do think that way. We found the genes, just go make a medicine, please. What's so hard about that? Chris Austin put something together a few years ago, which I loved. This is his view. It's completely impenetrable. That's if you know exactly what you're doing, that's the path one would take to make a medicine. So it's a little hard and a little complicated to get there. How can we simplify this? So I think we are in a situation now where we've got an ocean full of opportunities, and we're boiling in the ocean, and we don't know where to start. Okay? I think there are some clues on where to start. My favorite place to start is interesting properties. It has properties where there are some people walking around with a type of mutation that decreases the effect of that gene and is protective. Other people walk around with a different type of mutation, and it goes in the opposite direction, disease susceptibility, both sides of the curve, right? And people in the middle are people in the middle that have. So nature told us that actually it's a dose-response curve. We know what will happen if we go up very high, and we know what will happen if we go up very low. We don't need to test anything in mice or in model organisms or else. Nature told us that happened. So these are great. From a drug discovery perspective, it de-risks the whole equation. You just go in, you find these type of genes. The problem is There are not very many of them. We haven't found very many of them. This is one, PCSK9. There's one called NAV 1.7 for pain that has those properties. I'd like to argue that actually some of the findings that we've got coming out today are going to identify more of these. So I'll tell another story. They call them genome-wide association studies. Those blue diagrams I showed you before with the green pictures, those are from Genomewide. So you look at the whole genome and you ask what genes are associated. I've been known to be very disparaging of some, as one of the people who helped get those off the ground, I've been very disparaging of some of the activities in those, not because they're not worthwhile, but because I didn't necessarily find them scientifically intriguing. And the one I've probably been harshest about is height. Because every disease study you do, people collect height. So it's not really like yet lots of work has been done on the genetics of height. In fact, probably more than just about anything else, because it's there. Because we go measure asthma, we got height. We measure cardiovascular disease, we got height. And so we just test it. So locus. 2008 went up to 20. A couple years later, 180. 2014, we got 423 loci for height alone. And the latest one in bioRxiv, we got 712 things for height. So one would think that there's plenty to work from there. Now, it's kind of interesting. Of all of those loci for height, how many would be good for drugs, right? I said we've got an ocean of findings here. How many of those 700? I mean, I'm sure if people go study a million more people, there'll be 400 more. But how many have the properties that we're looking for? 4. This one pathway that we know, and they're all doing the same thing. You have mutations on one side that cause achondroplasia. You have mutations on the other side that cause extreme height. And the GWAS findings are right down the center that look just like HMG-CoA reductase in the middle. To generate the next generation of medicines. So this is the situation with achondroplasia, that you actually have a bit of biology, and there's an experiment of nature right there. So I used all this first half of my talk to set this up and give a genetics lesson, which sounds like it has nothing to do with large cohorts. Because the question is, if you buy this argument that we have lots of information, we have the tools that we need, and we got to figure out which ones to go after, how do you get the height type information at scale? Right? That height type information, I was really disparaging, but actually came out of 700,000 or 800,000 people. Because you could. How do you get that? And how do you get those type of findings where the gene first looks like it's operating on type 2 diabetes, but actually is for a related phenotype? You're not going to get that out of your type 2 diabetic patients. The only way I can see to get the information that lets us find these type of things is cohort. From a drug discovery perspective, that's why this was the third leg of the stool, where you've got genetic findings, genetic tools, and cohorts, because the cohorts are the things that are going to enable all of these findings to come through. Otherwise, we're just stuck looking paralyzed at a pool of information that we don't know what to do with. So Will mentioned that the All of Us program, I think many people are reasonably familiar with that program. I'm on the advisory board for it, and I did chair the initial version of the genomics working group to decide what to about All of Us. We can have comments and questions about All of Us because there's no data yet, right? I'm going to talk about some other programs and then tie it back to what All of Us will bring, so that we can talk about some data in real life. But the All of Us uniqueness is it's the largest to date. It's meant to be 1 million people. It's patient-centric, so it's not there's a big philosophical difference that every study that I bet people have been involved with here are designed by the researchers for the researchers. We design them, we say what we're going to collect, we say what we're going to test, and we go get it. This is sort of the idea is go the other way and have the participants decide what they're going to give, how deeply they're going to participate, levels of consent, how they're going to play this. It's a really different twist on that. We'll see how that works. And it's I think, and the diversity is where I was going with that. Most of the studies that we've got, and I'll talk about a few of these and highlight a few more, for various reasons, most of the large cohorts that are out there today, and there are a lot of them, are not at all genetically diverse. They're Western European Caucasian by and large. And so we understudy minorities, and all of us has gone after more diversity as an attempt to try and rectify, at least in part, some of the basis of that. So that's all of us, and we can come back to that and talk about what it may deliver and where it fits in the equation of all these others. But I wanted to talk about two others. Where these things are going. And both are in the UK. Part of that's my own familiarity because I was there when they were getting started and getting initiated. But both have been going for some time. The first one is called the UK Biobank. It's 500,000 people, age 45 to, it's on there, 40 to 69. It was started in about 2001, and see if you could get a grant for this. I was on I reviewed the grant for this. And the boldness of that grant was, we're proposing, we need 60 million pounds, which was a lot at that time. We need 60 million pounds to design a study that's not even going to be interesting for 15 years. That was the proposal in the grant, and they got it funded, which is pretty impressive. Not sick today, they're not type 2 diabetics, they're just individuals in the population at large. They will have their conditions over some period of time, and we'll watch. And that's exactly what they have done, and that was the design of that study. 500,000 people, collect the baseline information as best you can, and go forward. Now, the UK is a pretty special place to do this type of study, and it's easier than the US because it has a national healthcare system. So all the diversity that I'm sure we're going to hear about in terms of harmonizing phenotypic information is a little easier over there. Just because it's one system, they don't have the mobility of moving from this to the other, and all the different practical challenges that we have in the U.S. But that's the Biobank, and so they collect. They've got. It's not perfect, and it's not as expansive, I think, as all of us. All of us is going to put wearable devices on people and kind of make it 21st century. Now, some of you here, I'm sure, will have been part of this. Others would be blissfully unaware. But about a year ago, I think it was last summer, Biobank finally said, okay, it's a big reveal. All the genetic information is going to be released to the community. This is a very open, transparent. Industry, academia, everybody gets it, right? You apply, you give a research proposal, you get the information. And then on a Friday, we're going to release it to everyone. So nobody went anyway. Literally, people were waiting for this. The programs were all just ready to be the button pushed. The first paper came out in bioRxiv, I think, in a week. So it was written before, and all they did was populate the tables and that sort of thing. It was really an exciting time. Now, you can see on the bottom here, so 406 papers already on that thing. So that really delivered. The prospective, hold on, just wait for it, and you'll see the results at the end, has been absolutely nothing short of. They continue to this day, that program. And it's not even perfect. I mean, that's sort of a passive study of just getting genetic information and some imaging data and some metabolite information on a sample that's already there. There's no active real participation or collection of that. But it's changing the genetics altogether. Now, I'll give you just a fun example of how this works in practice. So I'll skip that one. I got a better example right here. So when I was still at Glaxo, just in Cambridge, from a professor called Stevo Riley, and I hired one of his postdocs. And his PhD program was to find a gene for coronary heart disease. And so now this is not 2001 I'm talking about. This is a couple of years ago, right? So his PhD program was to do that, and it took him 5 years to finish his PhD, did a nice job, got a big paper out of it at the end, and found a gene that he was interested in. You can see it on the It took me 5 years. I had to assemble 50 cohorts of individuals because no one had a biobank type program like that. So I just phoned up everybody in the world that had data that were relevant to the question I was asking, pooled them all together, figured out how to harmonize them, and made a discovery. Now I got the biobank data. So that on the left is his 5 years and 1,500 emails. Biobank data came out 1.5 hours and 3 emails. And one of those emails was to me, to ask if he could present the data. So that's pretty impressive that cohort of 500,000 people transformed, this is just in recent times, that level of genetic research. I mean, it really is transformational in the type of approaches that can be made. So beautiful, he got the job. It was a really good job talk. And now what we're seeing out of that I think is going to have a lot of impact in medicine and in drug discovery, and people call them polygenic risk scores. So just so you know, this is something that's coming out of these type of databases and large cohorts. And they go like this. I told you about those genome-wide association findings, 712 loci for height. Each one of those is tiny, right? In fact, there used to be, at early days in type 2 diabetes and obesity, it's about as good as BMI, right? So none of them are individually very predictive. But actually, if you take the findings today and you look at all them, what this picture on the right is showing, if you take just measure all of our genotypes here, right? And we each have slings and arrows of outrageous fortune, and you sum the low height loci with the high height loci, and you just add up our score for what nature gave us. We don't have any big mutations, but you add them all up. Those individuals have as good of prediction as if you had a Mendelian gene. You add them all up. So the polygenic effect on the extremes could work as well. If you think about it, it's just bad luck. Take the height example we've got. Those that are way out on the tail may not have any major gene mutation for height, but just unfortunate, got the stack of all the little ones that summed up and put them 3 standard deviations below the mean. And you take those out, and they're now predictive. So we've stepped out of the Mendelian case of having a 1-point mutation that has a really big effect. You got no big effects, and we've got information that we can use. Now, I think this is really, really exciting for where we take precision medicine and others, because where these findings lead us into pathways on the drivers, I don't know where it's going to go, but this is a super exciting area. In Mass General, I think. So that's the UK Biobank. So I wanted to tell you a little bit about that. Now I want to tell you a little bit more about another UK program, which is called Genomics England. So this is unrelated to the Biobank program, with a very, very different remit that I personally didn't think would 4 or 5 years ago, not longer. And the idea there was a different one from, this was a bespoke, let's go for rare diseases and let's go for cancer, specific in the UK cancer. The cancer program in UK Biobank, or in Genomics England, is because the nationalized healthcare system has some simplified resources that we don't have here. And part of that is they have a whole bunch of centers for rare diseases that are concentrated. So all the patients, if you have PAH, there's only 7 places you go in the whole country. If you have pulmonary arterial hypertension, there's only a few places you'd go. So easy to identify the patients. And what the Genomics England has done is just collect them all up and get DNA sequence on full genome DNA sequence, not just genotypes, on all of those individuals. And they're collecting them up, and they're up to about 100,000 individuals now. So they've got probands. It's a very, very different collection than just a population cohort, and it's rare disease centric. And coming along really, really well in terms of their sequencing and bringing together things in the UK. Now, in terms of the disorders that they have collected, I show this to make a point in just a moment. There's, I don't know, 31 different diseases on there. What they've kind of oversampled in Genomics England is cognitive deficit, right? Undiagnosed neurological diseases, with the idea of trying to improve that particular setting, where same in the US with the UDN, there's so many cases of heterogeneous etiology that we don't know. So there are a lot of individuals in this Genomics England for whom all we know is there's intellectual disability or cognitive deficits of some sort. Don't know the disease, don't know. Moment. So you go in, I work at a rare disease company, and this particular resource is just like the UK Biobank. It's available to all, which is something maybe we come back to because it's really powerful that we don't have distinctions between industry and academia on these type of things, at least today for basic research purposes. And the first things that we see, this will surprise none of the physicians in the room, but the first thing that one sees going in there is these are different genes on the left, all of which have treatments available for those particular lysosomal storage disorders, PKU, et cetera. That you've got definite undiagnosed or misdiagnosed patients sitting in those collections. As we go forward, I think we're getting the DNA sequence on the UK Biobank now, that 500,000. We're going to see them there. Different dynamic because they're older, so selection might have weeded them out, but we're going to see these. On every collection, all of us is going to see them. We're going to see people for whom the diagnosis was incorrect or just missed altogether, and they're available treatments. It's a win for everybody if we can get those the treatments to those patients. So that's England that we think is very interesting, and again, the clinicians in the room probably will see this as a no-brainer. I think it's an interesting but unexpected route to precision medicine. We're at BioMarin, we're interested in PKU, and we have treatments for PKU. So we're interested in how many PKU patients were in this sample. And there were a number of PKU patients, and the bottom two were probands. So these are probands, they're being treated for phenylketonuria. That's a very, very old genetic disease, and there are various treatments available for that. But there were 4 who weren't brought in because their children had cognitive deficits of some form or another that were undiagnosed. They just happened to be sequenced because they were the mother of someone who had a condition. Otherwise, we wouldn't have gone after them at all. So these children, their children, are being tested for every battery known to man. They got metabolomics coming in. And the interesting thing from a partly precision medicine, but just a cohort effect, is there is nothing you could ever do to find out in those kids what caused their disorder. Because they don't have a genetic mutation that's causing the disorder. They got it maternally from undiagnosed mothers before they were born, in utero. So their mothers had undiagnosed PKU, they had hyperphenylalanine, they passed it on. There's absolutely nothing you'll ever do in a study like this to find out what was wrong with the child, because that defect was done before. These type of, so it actually helps subdivide that part of the intellectual disability, but not in the way you would have ever thought, not in finding their genetic etiology, but actually finding the familial form of that. I think this is really fascinating. I wanted to draw out this particular sort of case study here, because I don't know where you are in Utah in terms of using the families, but the families have to come back here into human genetics. We've walked away from them. We do these big population, the field, I mean, they've got these massive cohorts that don't generally don't have family members unless they're just by serendipity. But the information, again, so that's the example. Where are we going next? So the UK has recently claimed they're going to move, so they got 500,000 in the UK Biobank, they got this 100,000 collection. They're going to go to 5 million and basically sequence the emerging population, the newborns for that. So I think this is really, really fascinating, because what they're doing is setting the bar for these cohorts. They're going to go to a population basis. We're going to get all the DNA information. Again, it's exciting because for them because they have a healthcare system that kind of automatically brings the clinical information in. That wouldn't be easy for us. Even if we could pay for the sequence, we'd have to figure out sense and how to do the ethics behind all of this. Research and a translational perspective, I'm really excited about this, because what happens next? If the UK does it, Scandinavia is going to follow, Europe, where do we go from here? And then things start to get really interesting, because we've got access to these massive populations where things get really interesting. We move beyond 500,000 into the all of us type scale of a million and multi-millions. Really, really exciting. More to come on this. This is a very clumsy slide, but to tell you, is it just the UK that has this and all of us in the US? And there's a Jeff Ginsburg and I think Francis Collins and some others organized a meeting last year, and there's another one just coming up next month, where they tried to identify who on the planet, any country, any cohort, any university or otherwise, has 100 specimens, urine or others, and we could follow them up. Who's got, just with those criteria, how many of those are out there? And they found 60 cohort studies, 25 million people ascertained today, 36 million planned, across 24 countries already. So it's not just a UK Biobank, US all of us thing. These things are out there. Now they're very, very advanced, but 25 million people already potentially there. They're here. And we're back to that situation. What do we do to translate those? So this is really exciting. And to let you know, to bring it back to the translation and the industry side of things, the fact that these populations exist and are valuable has not been lost on industry. A couple of them are here. Amgen bought Decode 4 or 5 years ago now. Regeneron has invested very, very heavily in this area and sequences everything they seem to be able to get their hands on. AstraZeneca's made, GSK recently put a big investment in 23andMe, these type of things. So there is financial support for the pieces. With the cohorts, with the genetic findings, with the tools, they're all there. And it's up to us to try to figure out how to really draw it together. So I'll end with what's not there. This is just my own perspective, or what's, even with 25 million people, no single population is enough. So even though I sit on the All of Us advisory board, they probably went 1 million people. Find attractiveness to this line of research from a translational perspective is not in the individual data sets, but in the collection of them. And for that, I think our colleagues, and you must have been very prescient in this particular meeting to bring together AI and the cohorts. That's got to be part of this equation, is a computational side of things on how to bring these data sets together, because that's really not going to be easy. I said a moment ago, we've got to invest in not just cohort studies, but in samples that either have families or consanguinity or anything that can amplify genetic signals. Our findings. This problem, it's not an area I work in, but I'm certainly a consumer of it. This challenge of harmonizing or bringing together phenotype information, I feel for the people that work in that space, because that is really, really hard clinically, especially in the US. And I can only imagine we've got all these disparate systems in the US, and then we want to link them with Finland or Japan or Taiwan, all of whom have their own settings and challenges today. So getting over that. And then finally, I don't think we can invest enough in the tools and infrastructure to bring this together. I just don't, I think if we all put the money that we had together, it wouldn't be enough to really make this happen and bring it in a way that's both for basic research and for translation as efficient as it can be. So having said all that, so I think this all brings together the precision medicine angle. I think the rare diseases, when we bring in the population-based, these cohorts that we're talking about right now, draw us naturally, as I've tried to argue, straight to common diseases, and we go rare to of that rather than the objective at the outset. I'll stop there and take any questions if there are any. Thank you.
Any questions from the audience? Alan, Dr. Morris.
Can you hear me?
Yes.
Without a microphone? Thanks very much. That was illuminating and convincing. I'd like your comments about a distinction that I think is not adequately made. What you described will clearly lead, I believe, to healthcare benefits, presuming that there's less emphasis on profit. Interesting in the opening paragraph, the right treatment to the right patient at the right price. I usually say at the right time. The right price suggests some other imperatives are driving that description. What you described won't lead to that, because that involves behavioral modification, primarily of clinicians, human decision makers. So I'd be interested in your comments about the distinction between precision medicine, and what you have described is just like the electron microscope. Any new way of looking at the world is extremely likely to produce new insights.
Yeah, thank you for that, because I think, and thank you for not drawing us into a. No, I think you're absolutely right. Where I have emphasized is more of a discovery, and tried to, thanks for suggesting it's convincing, and tried to emphasize that there is scope there for that. I do think there is some elements of precision medicine that maybe I didn't draw out that could come from this. So take the height example, right? And so nature has told us that there's a handful of genes that influence from achondroplasia all the way up to excess height. So anywhere on that spectrum, actually, if we drew people with those genetic mutations, we could alter height. Tall, taller, short. That. And another example, which I should have brought out of here, again, you're absolutely right, this was focused more on discovery, but I should have brought out of here are, I think, some really surprising findings that are coming out of rare diseases into the common diseases. So Gaucher disease. traditional lysosomal storage disease, rare Mendelian effect. Big genetic effect. That same gene that causes Gaucher disease causes 10 other mutations in that gene that are less severe, have an odds ratio of 10 for Parkinson's. So I accept your general challenge on the whole thing, but I think I would like to think there are situations where we could get the right treatment for the right patient genetically out of this. But go ahead.
Yeah, what I was getting at is the compliance of clinicians with evidence is so low, it's embarrassing. So for
I see.
personalized medicine to be achieved, the right patient at the right time.
Yeah.
And that's a very different domain at the clinician-patient encounter and the decision-making than what you've described. And, you know, I admit right up and now have something that looks like it's promising. I just don't understand that very well. But I do understand that this new information will only overburden already overburdened decision makers. Get the doctor to make the right decision. I just want to make that distinction between this very exciting stuff and precision medicine.
Thank you. I missed the question. I think you're absolutely right. In fact, I would open it up. Are there other suggestions here? Are you guys working on this to try and bridge that gap? Because I hope what you're not saying is, gee, it's so hard for the current climate of physicians, just ignore it. Because that's not what we need to do, right? We need to crack this problem. And it's
I also find that those super interesting analyses because they sort of give almost a null model for what you're after. So what then are all of those other weakly additive markers? Do you think that's cryptic stratification?
I think they're generally, it could be. He's asking if they're artifactual in some way or another, or I think some of them could be. I think generally they're that is so far away that a slight defect in something that isn't directly relevant to skeletal growth or development at all just has a tiny effect. It's the polygenic model, is what I'm assuming there.
We are understandably anthropocentric in this discussion, but we also know that a very high percentage of our genes and genomics are similar in animals. They are less concerned with privacy and legal issues. So is it possible to use animal sampling to tell us some information, or at least to allow us to focus a little better to make some of this easier?
World, and it was a revelation for me that in that case, when you start with you already know the human defect, I would have thought you just skip the animal models, but actually the animal models are really important, really, really important. So you go from man to the animals and then back to man. That is exactly how it works. So you understand what to measure and the effect size and the better physiology of really what's going on. So I think definitely there's animal use there. What I don't know, and maybe you've got an idea in your head, is this problem of having so much how you crack that with any organism, because it's so hard to start with. That's why I liked the height example, because just by looking, it narrowed you straight into a pathway. And so, and I got to think that when you see it once, there'll be others that do the same sort of thing. Maybe the animals could do that. Maybe it's some fancy editing study in cell models could help weed things out. But I take your point. It's really, really hard in men.
Thank you for your talk. That was very interesting. So the majority of the conversation has been about precision treatments, but I'm wondering if perhaps the UK has any lessons to teach us about applying these genetic methodologies to precision surveillance or precision prevention.
If they do, I don't know. I don't know if there are lessons yet on that. I think what the UK has been really, really good at and continues to be good at is leading the way for research I don't think they've, in practice, had a genomic medicine remit yet that would have impacted prevention. Now, they may. I don't, but I don't think that they have just yet.
Lon, I'm with the Precision Genomics Group at Intermountain Healthcare. Thank you for your talk. My question is a little bit different because I think you mentioned that one of the major problems is that there's plenty of cohorts out there. People want to sequence hundreds of thousands of people. One of the challenges is how you combine this data, right?
Yeah.
How do you, what are some of the major challenges you see and some of the compatibility issues that you see in terms of putting the data and the structures on these cohorts together so we can do bigger research?
Yeah, I'll tell you what now again, I don't work at that information challenge. There'll probably be others in this room that can weigh in on that. But here we've got completely different storage documentation of clinical phenotypes. We've got the longitudinal storage of that information goes, because we move in America, so we change healthcare provider and all that. We've got to get it together, and it seems to me you could make that really crude and blur everything, but then you're throwing out all the phenotypic detail that you actually need. So what we need to do is preserve as much of the clinical sophistication that we can and find a way to link them together. Now, others may be able to speak to that on a practical basis much better than I can, but we've got to find a way to do that without just crudely making big buckets, or else we've got this exquisite molecular information and this really blunt clinical information. We won't find anything. I think some people want to respond, which would be really helpful.
So very nice talk, thank you. So taking Crohn's disease for an example, there's a huge environmental component. Are any of these UK or other large cohorts including environmental factors?
They are, the UK ones are kind of passive, right? So it's kind of what is already there in the GP's computer or files. But it's a good call out because I always emphasize the genetics because I'm a geneticist. That is not to minimize or otherwise discount the environmental side. And so I think the UK Biobank, what they're doing is they've got this sample of individuals. It was an elective participation. And then now as they can get scientifically interesting questions and funding, they go back and recontact. And they collect whatever information they can. But it's not optimal that way because they didn't get it at baseline. They just got whatever they had.
Alan Morris again. Thanks so much for the break. I'd like to ask you a question I think Darwin would ask if he were in the audience. He understood that evolution produced changes, some of which conferred advantages and some of which were just riders, some of which were just pari passu along with something that developed advantages. So the question I think he might ask is, what does it mean to have 50 genes associated with height when we have no idea what mechanisms might be operative? My understanding of machine learning is the computer scientist would come to a domain expert and say, I've got all these associations for all these nodes, which are reasonable and which are silly. So you know right off the on how we might be misled or how we should proceed to evaluate the associations that may or may not be meaningful.
So you're recapitulating exactly the challenge. I completely agree, and I think that was part of the question we had before. Or are they just passengers in the course of evolution? I don't know. That's why we're paralyzed with all these findings. I like the height example because you take 700 potential different sites, and at least there's one mechanism pointed us to a pathway. Now maybe we can use AI and others to do that for other disorders, but we have to weed out the 48 out of 50 for Crohn's disease or for height or whatever you want that are too distal to the disease and not really going to matter if we're going to translate these. So your question or your concern is the same one I'm challenged with. How do we make use of this breadth of information?
Hi, thanks, Dr. Carden. Barriers that are still being faced here. And what you didn't mention, I'm sure there are others that you could, it's basically what I would call error, generally speaking, error in testing, as pre-analytic, analytic, post-analytic error. I assume all of these different databases are generated from different testing systems. Do you think start blending these databases together?
Yeah, thank you for raising it. Dramatically so. So let me answer it in two ways. On the genomic side, right, that community, since because there's sort of a truism. Every major finding we've ever made was a false positive first, because it was always genotyping error or something. Really subtle findings could make it. So from the molecular side, there's been really close attention to be as exquisite as possible. So you have this really sharp knife that's good, that is really, really good, and you're linking that up with phenotypic information that we're mixing and matching and comes from different sources, and I think there's the weakness. Right? It's not the molecular side. It's more sharp than it needs to be to get there. So yeah, I don't even know if we know the magnitude of the effect of that the errors have just yet on this. This is why I say you couldn't spend enough money to
Wonderful presentation, Lon. It is it's my understanding that the VA Million Veterans Program has collected samples on more than 500,000 veterans. I was curious to get your thoughts. MVP.
Yeah. There is a lot of coordination between that, and in fact, before I was on the advisory program for all of us, they were synchronized in terms of design and study, including the UK Biobank too. So sitting on that initial All of Us program, you had Rory Collins, who chairs the UK Biobank. It had, I forgot who it was now, from Million Veterans. They had all the people so that all of us could draw on the learnings from Biobank does. Data access is some, consent, return of results. These are all really live and challenging questions, and each one set it up differently. So I think, and I don't know that there is a single solution to this. So yeah, there's an attempt to try and learn from them and bring them together as best they can.
Dr. Morris, you have the last question. You stand between us and the break, so a short one for your third one is perfectly appropriate.
Well, I ought to speak for a long time because this is a health maintenance organization. They've got a lot of I'd like to follow up a couple of comments that link to context, epigenetics, and era. Okay. All of the machine learning, the clinical phenotype, that are acquired for other purposes. Yeah. So you've got a universe on which the machine algorithm is operating, but the universe has two major problems I'd like your comments about. The first is it's clearly incomplete. There are many things driving clinical decisions and patient decisions that are absent from the universe, so the machine learning can't deal with those. And the second is it's full of noise, both random and systematic. Yep. Could you comment about that and how misleading that could be?
A little bit. So on the noise, right? Let's split that up. You hit it exactly right. Systematic versus non-systematic. So one lesson that I learned out of 23. Them some saliva and they'll do some genotyping for you. And all the clinical information is self-reported on the internet, right? So one might think that could be pretty clumsy information, pretty erroneous information could be, you could be a joke about it, and missing data as well, because you choose what you want to complete and not. And so that could be laden with all ones. There's all kinds of noise and error in there, but as long as it's not systematic, sample size trumps it. So you get bigger and bigger and bigger and bigger, and it just it adds noise, dilutes the signal and all that, but it's still there if you can get big enough. So I think, actually, if we draw them together, the non-systematic bias, we might just get past. It's the systematic bias. I think we just
Please join me in thanking Dr. Carden for a fantastic lecture. Now we have a 20-minute break, and we'll start promptly at 10:30. Thank you.
If people could start to take their seats, we're going to try to start up again in about a minute. Okay, great. Thank you. Well, welcome back. I'm George Contreras. I'm a professor of law here at the University of Utah and have had the privilege of being one of the co-organizers of this event since its inception in 2000. Follow up on some of the really thought-provoking material that Dr. Carton told us about in the last session. And so this panel relates to ethical, legal, and social issues arising from the assembly of large cohorts for precision medicine. Laura Lyman Rodriguez, who is the director of a division of policy, communications, and education at NHGRI, sends her regrets. She was one of our scheduled speakers on this panel, but a last-minute emergency came up at home, and she's not able to join us. She extends her regrets. However, we do have three outstanding panelists who are going to explore these issues, each from a different angle relating to the ethical, legal, and social implications of these types of research in these large cohorts. The first of whom, and I'll just introduce them each, and then in serial we'll have them come up to the podium and make their presentations. First is Jim Tabery, who's an associate professor in the Department of Philosophy here at the University of Utah. He also holds an adjunct appointment in the Department of Pediatrics and the Department of Internal Medicine. After Jim, we will have Kimberly Kaphingst, who is a professor in the Department of Communication here at the University of Utah and also an investigator in the Cancer Control and Population Sciences program at Huntsman Cancer Institute. And the endowed chair for pediatric clinical and translational research at the University of Louisville. So with that, I'll ask Jim to, oh, there he is. I'll ask Jim to come up and, was that me? I think it's my lapel. Oh, okay. So, about 20 minutes, and then we will have ample time for Q&A, where all of us will go up to the table and field any questions that you might have. So, Jim, up to you.
Still live. Would you rather me just use that? I'm going to start by showing a movie. It's only about 100 seconds, so it's going to be brief. But here's the thing, though. It's interactive, okay? There's going to be a bit of a scavenger hunt here. And what I want you to do over these next 98 seconds is keep track of, so you're going to need a pen and paper, keep track of every time you hear any of the following terms: gene, okay? So keep
We are one nation, one people. When called upon to give from within, we come together and here we are fearless. What lies inside all of us is more than data. It's life. It's more than insight and medical research. It's vision and honor. next great breakthrough will be found in each and every one of us. And what we find there will unlock mysteries, heal the sick, and eradicate disease. We ask for 1 million individuals to come forward and stand on this landmark in history. We ask America to do, once again, what she has always done. Lead the way forward. We're one nation, one people, but all of us are different. And it's those very differences that will lead to answers for generations to come.
Now, no cheating. If somebody next to you is checking their email, don't give them the answer. Okay, so what I want to talk about today is the idea that these large longitudinal cohorts, which on the face of it look like they're just epidemiological decisions, or it looks like they're ethical decisions, or it looks like they're recruitment decisions. And what I want to convey is actually they're ethical decisions, okay? And by that I mean there are ethical implications of these epidemiological and of these recruitment decisions that are made. Okay, so I think first just a little bit of context is helpful. From Dr. Carden about the UK Biobank. I think it's worth keeping in mind though that large longitudinal cohorts come in all shapes and sizes, and it's a very old concept, right? Probably the oldest and most famous is the Framingham Heart Study, okay? That goes in Massachusetts, and they were interested in cardiovascular health, cardiovascular disease. It was a response largely to FDR dying of cardiovascular disease, and they wanted to do something about the epidemic, and he was the galvanizing case. Framingham gets set up in response, okay? That was planned to last 20 years, right? When it was done, though, when the 20 years was up, they decided to recruit the spouses of the people who were enrolled. They also recruited the children. Of people that have been enrolled in the Framingham Heart Study. It remains today sort of one of the gold standards of what a large longitudinal study can look like. But it started out as a very sort of odd thing. About 5,000 people from one town in Massachusetts. I'll also just sort of say, briefly, the Dunedin longitudinal study is a nice sort of analog to Framingham. And enrolled them into this longitudinal study. So most of these people are now in their 40s, and they've collected just a wealth of environmental, social, behavioral, and genetic information about everybody who was enrolled in that. Sort of opposite extreme, you can get these really, really massive studies like UK Biobank and All of Us. Here you're trying to get lots and lots of people, right? You usually want to be able to say, in some sense, it's nationwide, it's national. Okay, because of they basically recruit people typically in hospitals or in healthcare providing locations because that's where you can find the people that sign up for this. And then in between, I think a sort of interesting one that was sort of in between, which our local Utah community knows well, was the National Children's Study. The attempt with that was to get 100,000 babies born. It was designed as a probability study. It was a probability sample where it was sampled statistically in such a way that allowed you to make inferences about the national sample based on the sample that was recruited into the study. It also was heavily leaning gives you kind of, when we're talking, I think it's worth keeping in mind, when we're talking about large longitudinal cohort studies, these things come in all shapes and sizes. So the first thing you might ask then is, well, what's right? What's the right way to do one of these? Right? You've got a lot that ever lived and get all data from them, whatever that means, over the course of their life, right, and then link up all those exposures and genetic differences and lifestyle decisions that they make with their health record, right, that would be the ideal. Right? I mean, that's sort of like the ultimate large longitudinal cohort study. But obviously, we can't do that. So then decisions have to be made about, well, if we can't do that, what are we going to do instead? And it's is there a particular trait that you're interested in? Are there particular causes that you're interested in? Are there particular population level phenomena that you want to address? Depending on what you're interested in, that is going to also just some more practical reasons. How much money do you have, right? Who can you interact with? Depending on the answers to those questions, that will end up dictating what are better and worse answers to these sorts of questions, right? So how many participants? 1,000, 5,000, you can go up to 1 million. Who are we going to include? Are there going to be undocumented immigrants? Are we going to have children? Is it just going to be a cohort of largely upper middle class white people that trust the biomedical research industry? Right? What is the sampling going to look like? Is it going to be a national sample everybody in a single town? Right? What is the focus of the data? Are we looking largely at genetic information? Are we looking largely at physical environmental information? Are we looking largely at social behavioral environment, right? Family environment? Lifestyle decisions, that could be another focus. Another sort of one that I didn't put up here but is worth sort of emphasizing is you have to make a decision about whether or not it's going to be hypothesis informed. So some large longitudinal cohorts start with a set of hypotheses up front about what they want to test, and then the sample is generated in response to what you need to answer that question, right? Depending on what question you've got, that's going to tell you how many people you need. Depending on what question you've asked, that's going to tell you what the sample needs to look like. As a cohort, as a resource that people can then bring their hypotheses to, right? And so what the first lesson I just want to say here is, in the epidemiology, that's going to tell you how to move through this, right? Something like the Dunedin study or Framingham, 5,000 people, largely wealthy white people that trusted research. It was in a town, right? And it got a lot of environmental and lifestyle data. Switch over to all of us, shooting for 1 million people, right? Talking about rolling children in, hope it's going to be diverse. It's a convenient sample, supposed to be nationwide, lots of focus on genetics, okay? No right or wrong answer here. It's what you're interested in. What I want to emphasize next, though, then, is these decisions have ethical implications, and they're the things that people need to be. The question, obviously, is who's going to benefit from the research? Decisions that are made about the study population, the sample population, impact what you can and can't infer about the target population. And if the target population is, people in the United States, or it's humans, well, then you need a sample that justifies that kind of inference, okay? And it's just a reality that depending on what you're interested in, a convenient sample won't necessarily let you make those inferences to, say, a national population. For some things, it's okay. For other things, not so okay. Right? What's the best use of scarce biomedical resources? This is in partly a sort of a study, Framingham, they had a very sort of specific idea about what they wanted to study, and they directed resources towards collecting data that would allow them to weigh in on a particular hypothesis that they were interested in. And it was largely environmental in nature, okay? Switch over to all of us, right? It is much more focused on genetics, okay? That's a decision that has to be made. It's not just about what does the science say. It's about what traits are you interested in? What population level phenomena do you want to address? And does it make more or less sense to put your eggs in this basket, or just how are you going to distribute your eggs? And then the last one that I'll just flag is, I think this, on the face of it, looks like sort of a no-brainer, but I'm about to suggest otherwise. Does the recruitment and advertising materials the answer should be yes, right? And the sort of governing principle of biomedical research is that it's going to be yes, okay? I would like to now indicate that for a pretty notable one, it's not. Okay, so let's just take a moment to look at the actual design and history of the All of Us research program. This was, we'll talk about the history. This was the vision of Dr. Francis Collins at the end of the Human Genome Project. It's something that he wanted to design when the Human Genome Project was wrapping up so that the genome the early iterations of it and the design of it were designed at the Genome Institute. And then subsequently, the groundwork for it was laid at the Genome Institute with programs that you may have heard of, programs like the Gene Environment and Health Initiative, or eMERGE, which was a big effort. Gene Environment Health was an effort to sort of coordinate genetic and environmental research, look for gene-environment interactions. eMERGE was an attempt to link up genetic information with medical records and draw the connections there. Okay, and the vision all along for those things was to set the stage for a national massive study. Okay, so the history of this is, and I'll just these are always massive genetic studies with genes in the driver's seat, and then people who are interested in the environment are sort of allowed to come along for the ride. The design decisions are going to be guided by genetic interests. If what they're collecting could be of use to you, environmental scientist, we're happy to have you on board for the party. But okay, that's what the history reveals. Let's just then look at the focus of the current All of Us program. There are 3 genome sequencing centers. That shouldn't be surprising coming out of this history. Is there an No. There is a genomics working group. We just had one of the, our keynote speaker was on it. Is there a lifestyle working group? Is there an environment working group? No. Let's look at the data they're collecting and the sample size. They are making it a priority to get blood right away. And when you look at the protocol, why are they getting blood? They're getting blood to get DNA. I'm in it. You answer a bunch of surveys. You say, well, yeah, that's me. There's a promise that smartphones and things like Fitbits are going to come online later. I can tell you, if someone gave environmental, social, behavioral health scientists $1 billion and said, go, this is not what they would be designing. They might be along for the ride, but this is not what they designed. Okay? It is a massive convenience sample. 1 million people, I mean, all you geneticists are chomping at the bit. This is going to be like the biggest genome-wide association study we've got. I get it. You're excited. But that's a decision that's made. You can get lots of genetic information from 1 million people, right? It is going to be very difficult to get substantive environmental information from 1 million people. They can answer questions on their smartphones. They can log into their All of Us app and say what they had for breakfast that morning, okay? But that is a very different thing. So, a massive genetic study. Now, back to the scavenger hunt. How many genome, DNA, genetics did you count in our video? 0. Here is just let's move through this sort of in a chronological way as if you were sort of first hearing about the All of Us research program, got interested in signing up and then did sign up, okay? Based on that video, you've got nothing about the previous slide. Here are some, I'm not going to pass these out, these are very valuable. These are All of Us research brochures. So if you go and sort of find information about it, this is what you're going to find. I'll give you a guess. How many references to DNA, genetics, genome are in these? 0. There are almost no references, you can find a couple, especially if you go on the website and dig a little bit. There are almost no references to these classic genetic terms in the history, biological differences, what's inside us. They're terms that are supposed to key to the geneticists, hey, we're getting what you want. But they're not cueing the people that might be interested what we're getting. There are lots of references in here to environment and lifestyle. Funny enough. If you enroll in the process, if you go through the process of actually signing up, going through the consent process, okay, you will watch a bunch of videos, look at a bunch of slides. One of those slides mentions DNA. In about a 45-minute enrollment video, I think something like 30 seconds is devoted to DNA. And when you turn to the participation, even once you've signed up, enrolled, a massive genetic study. Okay, let me give you an example of this. I took this picture when I went and enrolled. So I enrolled online, okay? And then when the All of Us bus, most of the recruitment takes but you can also enroll when the tour bus comes through town, right? So I went to the tour bus when it came through town. I was enrolled. This is the room where I gave my blood. When you walk in, the first thing you see is this sort of big image. And I think what's nice about it is it sort of captures, right, it's giving you a visual taste of what All of Us research program is about. Can anybody see what would be a cue that this is going to be focusing on DNA? Way down there in the bottom. Lots of stuff on environment, lots of stuff on my health, lots of stuff on my house, on the sort of physical environment that I live in. The arm out of the way that they're about to pull the blood out of you from to realize, like, oh, maybe they're going to get some DNA. Now, if I turned around, there was a sheet there, because they fill like 8 or 9 vials of blood. And when you turn around and look at the sheet of what those color-coded vials coordinate with, it's for a bunch of DNA studies. So here's, wrap this all up. How should we use scarce medical resources, biomedical resources, to design these big studies that we hope is going to improve health and wellness in the nation? I think it's a complete legitimate debate you might have about, does it make more sense to dump billions of dollars in a study that's going to track 100,000 kids from birth into adulthood and collect vast amounts of detailed environmental information from them as they mature? Into a massive genetics study, okay? That's a debate worth having. But you can't have that debate if one version is actually not showing. That's not okay. Thanks.
foremost, thank you, Jim, for the really excellent introduction to our session. I'm going to be speaking to a number of issues that Jim brought up from the angle from a communication scholar. So how do we think about recruiting a broad population sample into these large cohorts, taking into account differences in potential communication strategies? As a really brief overview, I am going to introduce you to the idea of health literacy, which I'm going to argue is really critical again in thinking about communication strategies and recruitment of broad population cohorts. I'm a data person, so we are going to look at data on knowledge levels about precision medicine and genetics and how those differ across population subgroups. And that. And then I'll conclude with a few thoughts on strategies that can help us in recruitment as well as informed consent across population subgroups. So beginning with health literacy has been defined by the Institute of Medicine as a degree to which individuals have the capacity to obtain, process, and understand basic health information. Literacy really for two reasons. One is I find it a really useful framework in thinking about a constellation of communication skills that people would need in order to make decisions about participating in something like a large cohort study. But the second is and to some extent across population subgroups. So this is the best available data we have. It's now about 15 years old, but it was from a massive study that was done by the US Department of Education, a household visit. Tasks. This is an analysis of the health-related literacy tasks. And what this shows is that about 14% of US adults, so that is the sort of purplish box there, fall into what's called the below basic group. And are likely to struggle with essentially all literacy tasks in the healthcare setting. An additional 22% of US adults, which is the white box there, are the basic group. So these are people who have the literacy skills they need for a day-to-day life, but again are likely to struggle with literacy tasks in the healthcare setting. So combining those two groups, you come up with 36% of US adults who are likely to have some struggles with literacy tasks in the healthcare setting. And I would argue as well in recruitment or considerations for informed consent with large cohorts. I will also indicate there on the right, that's a sort of hash mark box. Those are the level of skills that we need generally for informed consent. So only about 12% of US adults are able to read most informed consent documents and make decisions. And so this is going to be a challenge for most adults. It's also important to note that levels of health literacy differ across, again, population subgroups. So what I'm showing you here is data by race. Hispanic or Latino, and American Indian or Alaska Native are more likely to have limited health literacy than adults who identify as white or Caucasian or Asian or Pacific Islander. So as we think about reaching out to diverse samples for recruitment into something like All of Us or a large population-based cohort, I think literacy is really essential to think about. I'm not going to show all of much more likely to have limited health literacy than younger adults, and that individuals with lower educational attainment are also more likely to have limited health literacy. There's a very strong correlation between education and literacy, I think, as you can imagine. So moving on to the data part, I'm going to show you a little bit about how health literacy impacts knowledge as well as familiarity. So these are data that were just published in the Journal of Genetic Counseling a So they recruited about 250 adults at Vanderbilt, Meharry Medical College, as well as the University of Miami. And it was quite a diverse sample. So about a third white, a third black, and a third Hispanic adults as people self different terms related to precision medicine, and then looked at differences by race and ethnicity as well as by literacy. So what I'm showing here are the proportions who said they were moderately There's not a lot of familiarity with precision medicine. The term precision medicine, these data were collected just under 2 years ago, and less than about a quarter of adults overall had thought they were familiar with that term. There was greater familiarity with genetic testing, although when you look at the open-ended data, a lot of people's familiarity in terms of criminal justice, not in terms of health per se. And you can also see that there was pretty low familiarity with the terms biobank and pharmacogenomics. I think pharmacogenomics isn't necessarily surprising, but people aren't familiar with the term biobank. What was interesting about this data is there were no differences by race and ethnicity, I should say no significant differences in familiarity. Split dichotomized participants into low health literacy or adequate health literacy. Individuals with low health literacy were much less familiar with the terms gene, DNA, genetic testing, and pharmacogenomics. And you can see that those differences are pretty big. So just looking at the term gene, the third row there, individuals with low health literacy, 34% were familiar with the term gene, compared to 69% among those with adequate health literacy. There are other data that have shown some differences by race and ethnicity in knowledge about genetics more broadly rather than specifically precision medicine. This was from he powered a study to look at differences by both race and ethnicity as well as gender. They didn't look at literacy in this particular study. But the differences they observed were actually not in the hypothesized direction in a lot of cases. Where there were differences by race and ethnicity. So starting from the top, the item when they're born, identical twins have exactly the same genes. You can see that most people in all subgroups got that correct, knew that that was a true item. But that black men and black women were more likely to get that correct than white men, who were the comparison group in this study. The third line, the middle row, our genes tell us which race we belong to, which they had as a false item. You can see that actually most people in all subgroups got that incorrect. Than the other groups. And then finally, the lowest row, there are different types of genes in different parts of the body. The answer, the proportion correct was somewhat better for that one, although you can see again, most people got that incorrect. But in that case, white men were actually the most likely to answer correctly. And so they had expected to see consistent patterns or differences in And just to mention from some of my own work from St. Louis, we have found that there's a strong correlation between limited health literacy and knowledge about genetics. So this is from a study of about 700 individuals who were enrolled from a safety net primary care clinic at the Barnes-Jewish Hospital in St. Louis. And what we found was that individuals with limited health literacy were much less likely to have high levels of genetic knowledge. So in other words, those with limited health literacy had significantly lower genetic knowledge. That was also true so that patients who were older, patients who were Black or African American compared to Caucasian also had significant of education. So in this sample, that was people with some college compared to less than a high school degree. Those with higher educational attainment had higher levels of knowledge about genetics. So there are also good data looking at attitudes, so not only knowledge levels, but people's attitudes towards something like a precision medicine cohort. This was a study led by Dave Kaufman, who's at the National Human Genome Research Institute, and was published a few years ago. This was a large population-based sample of about 2,600 adults who did a survey where they described at that point the planned should this study be done, as well as if it is done, would you participate? And this slide is showing the proportions who said the study should probably or definitely be done, as well as if it is done, I would probably or definitely participate. So the good news here is that actually there was broad support for the precision medicine cohort. Across all subgroups, people broadly supported, at least 3/4 of people said this should probably or definitely be done. And there were actually no differences by race and ethnicity in there. Where we did see a difference, or they saw a difference, was with educational attainment. So adults who had less than a high school diploma were significantly less likely to support the study being done at all compared to those with a college degree. You'll notice also if you compare the personally would participate in it. So comparing just the top row there as an example of people who are white or non-Hispanic, 79% said, yes, we should do this study. But only 53% said, yes, I would participate. So the study should be done, but other people should participate for at least a sub part of the population. There were some minor differences here by race and ethnicity on whether the person particularly would participate. But again, the bigger issue was with educational attainment, so that adults with lower educational attainment were significantly less likely to say they would participate personally. And you can see on the right there on the lower part of the slide, less than high school said that they personally would participate, compared to 60% with a college degree. University in St. Louis, not looking at attitudes or intentions, but looking at actually what people agree to if you ask them what they will share. So this was about 2,000 men at the time who had consented to participate in a prostate cancer cohort. And the way the cohort was described is that they were collecting biospecimens from men who had been diagnosed with prostate cancer. They also, in the consent process, asked, is it okay if we use your biospecimens in the future for future research related to prostate cancer or not? And is it okay if we attach your PHI? And I should say we didn't actually use the term PHI. It was explained that it was protected health information. So I guess the good news here is that most of the men were willing to have their The not so good news is that very few men allowed PHI to be linked to their biospecimens. So if you compare the middle groups here, those are the people in the middle, yes, no PHI, means yes, you can use my biospecimens, but no, you cannot use my protected health information. Those much smaller bars there on the right are people who said, yes, you can use my biospecimens, and yes, you can link my PHI. When we compare differences between black men and white men who had agreed to participate in the cohort, black men were actually more likely to say that their PHI could be linked to their biospecimen, but they were also more likely to say, no, you can't use my biospecimen. So then in the last few minutes, what I'd like to do is think about, so what do we do about all of this? So we know that health literacy is likely to be a substantial issue as we start to think about recruitment into large cohorts, particularly if we want a diverse cohort. And in thinking about this, I've really drawn on the work of the Institute of Medicine workshop on informed consent and health literacy. And I think some of their thinking is not only relevant for informed consent per se, but also approaches to recruitment. So I'll highlight a few things. So one is what they call simplify, and I will argue is actually plain language or clear communication, but also the importance of considering alternative formats, so not relying so much on print as we have a tendency to do in recruitment as well as informed consent. So clear communication, or what is often called plain language, I think these principles are fairly straightforward, and many of you likely have seen them. Writing to one main idea per paragraph, clearly organizing your information, using descriptive subheadings,